Good evening, friends. In the last period, we have seen the chapter called as biotechnology. And after seeing the biotechnology, we have seen the bioethics, we have seen biopiracy, we have seen the meaning of each and everything. The chapter was very important on the basis of the final exam. And after that, on the Monday, we are going to conduct one test, that is the test number 1. See, I have completed 2 to 3 chapters, and on that, now we will see the test on each and every, what we can say, the chapter. So, in the forthcoming 3 days, only we are going to see the MCQ test. If you see the outline of the test will be conducted of 25 marks. The test will be MCQ. And after that, after solving the paper, I will put the answer key. Just remind yourself, make the tally of the question and the answer, and be punctual, be honest, be loyal on the test, and make the evaluation of the test yourself, that how much you will get the marks. And after that, we'll see, I will ask the students who will be above 23 marks, who will get out of out marks. I will ask the student to give their numbers, and I will display it in the next chapter, in the next period. Friends, after seeing the biotechnology, we have seen how the gene therapy is done. We have seen how the transgenic plants are produced. The forthcoming generation will be totally of the hybrid plants, transgenic plants, how they will be produced, how the yield of the crop will be increased for the increasing explosion of the population. Nobody should be hungry. Our main motto is there of the biotechnology. What is the motto? The motto is that everybody in India should get the enough food. Nobody should die due to the malnutrition. Everybody should get the proper diet, because everybody has a right to get the enough food, and the food should be of the quality. That's what we have seen in the biotechnology, and the forthcoming years will be of the biotechnology. Because to fulfill the demands of the overcrowding population of the whole world in the year, that is the 1960 to 1970 is one the decade, and that decade is called as the decade of the Green Revolution. And Swaminathan from India, Norman Borlaug on the world basis. These 2 names are there, by whom the whole world is getting the enough food to eat. Because when revolution was there, that is called as the Green Revolution. After seeing the whole, what we can say, the clear concept of the chapter, now we'll move towards the next chapter, that is called as molecular basis of inheritance. Again, this chapter is very, very important on the basis of the final exam. First of all, you see what is the meaning of this particular chapter, that is the molecular basis of inheritance. Means what? On the molecular basis, we are going to see how the inheritance takes place. And we know very well the meaning of the inheritance. When the characters they are transferred from one generation to another generation, that is called as the inheritance. All the characters which are there, they are called as inherited characters. Suppose, if we take the example of the inheritance, we have seen during the last chapter, that is the inheritance chapter, mental basis of inheritance. What we have seen that? In that particular chapter, we have seen how the scientist Mendel, Gregor John Mendel, how he have worked on the pea plant and how the inheritance is there. Yes, the transmission of the character is there from one generation to another generation. This chapter is totally based upon that particular fact. Now we are going to see what is the actual fact. That is, the year from 1928 to 1952, these 20 years are very, very important. The scientist called as Friedrich Miescher, he first of all discovered the DNA. Before 1928, nobody was known about the DNA, nobody was known about the RNA, nobody was known about the protein, nobody was known about the enzymes, etc. etc. This was the fact. But after the scientist or the botanist like Miescher, he have conducted an experiment. Further, many scientists were there, such as Griffith was there, Avery McLeod and McCarthy was there, Hershey and Chase was there, and there are various types of, they have done the experiment to show that the genetic material was DNA. Means how the characters they are transferred from one generation to another generation, and to make the transmission of the character, which material is present in the cell, that is responsible for the transmission of the character, and how the characters are transmitted from one generation to another generation, or one cell to another cell. The world was unknown about it. This scientist called as the Miescher, first of all, he have discovered the DNA. From which cell? For the DNA, what he have given the name, he have called it as a nuclein. Okay, which was acidic in nature. Which was acidic in nature. So it is called as deoxyribose nucleic acid. The acid which is present in the nucleus of the cell. Before DNA it was called as a nuclein. And how he could found the nuclein, how he have discovered that this is a nuclein, from which cell he have discovered. The Miescher he have collected the pus cells from the local hospitals many pus-cell bandages, and after that by using the salt solution he have collected that particular pus cells while he was doing the study on the WBCS. After collecting that pus cells, he have made the solution alkaline. And after making the solution alkaline, he have found that or some solutions or the content of the solutions, they were separated from each other in such a way. He found that some threads were there, or thread-like structure was there. And furtherly he tried to find that what is that structure. After some days he found that the threads were nothing but the chromosomes, and on the chromosomes the genes were there, and the genes are responsible for the transmission of actual characters. Means the genes are present on the thread-like structure called the chromosomes, and the chromosomes are made up of DNA. This is the DNA. And a final conclusion was there that the genetic material which is present in the nucleus of the cell was the mixture of many contents. Somebody called it as a DNA, somebody called it as a RNA, somebody called it as a proteins, somebody called it as enzymes. But what was the actual conclusion? Inside it, the conclusion was that the genetic material is nothing but the DNA. And keep in mind, the DNA, they are rich in phosphorus. They are made up of the phosphorus molecules. They do not contain the sulfur. Sulfur is present in the protein coat. Protein coat. Remember it very well. It will help you in the next period or in the next experiment. And always the DNA molecule or the DNA molecule is also called as a genetic material, which is responsible for the transmission of character from one generation to the another generation. This is the actual fact what the Michel was found. But the whole world was not convinced about it. And many scientists were there. I will tell you the name of the scientists. They mainly shown the 3 experiments. What were the experiments? Number first was Griffith's experiment. Number second was Avery, MacLeod and McCarthy experiment. And number third was Hershey and Chase experiment. Now, what was this 3 experiment we are going to see in short? First of all, I will deal with the Griffith's experiment. The scientist called as Griffith, he worked on the 2 strains of bacteria, that is S strain and next was R strain. And these 2 bacteria have the different strains. What was the name of the bacteria? That is, he worked on the bacteria called as Streptococcus pneumoniae. Streptococcus pneumoniae. is a one type of bacteria. That is called as Streptococcus pneumoniae. That is the cocci form of a bacteria, which causes the pneumonia in the mammals. Now somebody will ask me the question that what is the meaning of pneumonia and how it is caused in the mammals only. Okay, that might be rat, might be cow, might be any other mammal, might be the humans. Mostly it is seen in the humans. And that disease is due to the different strains of Streptococci, Streptococcus pneumoniae. These bacteria are very, very dangerous. Directly they infect your lungs. Directly they infect your lungs. And whenever you are unable to respire, means the alveoli or small balloons which are present in the lungs, they are unable to store or restore the oxygen. Automatically your body will not get the sufficient amount of oxygen, and if the body will not get sufficient amount of oxygen, automatically you will be energyless, and finally the death will be there. That will be fatal to your body. But mostly it harms your lungs. Means some liquid will be produced, that is the watery liquid will be produced in the lungs, and the lungs are unable to respire. Very simple. And that type of disease is called as pneumonia. And these Streptococcus pneumoniae, it will produce the large amount of colonies in the lungs. If the infection is not controlled in the time, they will produce more amount of colonies, and finally it will be fatal to you. That is about the pneumonia. What is the strain? S means smooth bacteria, or the bacteria which has the outer coat smooth. These are virulent, pathogenic, fatal. It's when such type of Streptococcus pneumoniae that will enter in your body, automatically it will cause pneumonia, and finally the death will be there. What about the R strain? They are a rough strain. This outer coating of the protein is rough, but this type of bacteria, they are not fatal. They are non-pathogenic, non-virulent. Understand? This is the difference between the two strains. And he made the experiment. Where is the cloth? He made the experiment. That is, the experiment is conducted in 4 steps. Number 1, number 2, number 3, and number 4. Let me tell you what will be the first experiment. And these experiments were conducted on the mouse, or you can call it as a mice or a rat. What was the first experiment? He injected live strain bacteria in the mouse. Live. Automatically the rat causes pneumonia and he was died. Next experiment. He injected live R strain. Live R strain. The mouse was not died, the mouse was alive. Because these R strain, they are not pathogenic. They are not virulent. They are not encapsulated. And yes, are encapsulated. Okay. In the third experiment, what he have done? He have taken the strain in such a way that he have used heat-killed strain bacteria. Means that bacteria were collected in a beaker containing water, and that beaker was heated, boiled, and after boiling automatically the bacteria were died, and that content was given to the mice. The mice remain alive. What was in the fourth experiment? In the fourth experiment, he have mixed S and R strain. How he have mixed? The live R strain was mixed with heat killed S strain, and this mixture was given to the mice, and it was found that the mice was died. Then he further collected the blood of the mice or content of the mice of S and R strain, and what he have found? He have found that heat killed S strain was heat killed S strain was converted into live S strain, and the mice was died. And from this he have concluded that something is there in which R strain bacteria, they are converted into live S strain, that is virulent, pathogenic. Due to this, the mice was dead, and he called it as a transforming material. And what was the transforming material? The DNA was a transforming material. This was the final conclusion of this particular